Hey champs of Examsnet, welcome to the series of chapter-wise discussion. In this session, we are going to discuss the chapter Anatomy of Flowering Plants. In the previous chapter, we have discussed the morphology of the flowering plants. Okay, so let's first understand what is the difference between the morphology and the anatomy. In case of morphology, we had studied the external appearance of different parts of the plants, whereas in case of anatomy, we are going to discuss the internal structure of the plants. Okay, so what are the components of the internal structure? Obviously, the cells, tissues, and organs, these are the components of the internal structure. They are arranged or organized to form the internal structure of the plants, right? So we are going to discuss about these organizations or the arrangements, right? Now, another important thing is that in case of morphology, it was not required to cut any part of the plant because the external features were clearly visible from outside. But in anatomy, how are we going to observe the internal structure without cutting any part of the plant? Obviously, we need to cut the plant parts, and the best way to observe all the arrangement of all the tissues and cells is to take a transverse section of the plant part, because the pattern or the arrangement of cells are clearly visible in a transverse section. Okay, so we will consider a transverse section. Now we will start right from the beginning of a plant life. We know that the life of a plant starts from the germination of a seed. So here we have a diagram showing the stages of germination. Although this diagram is not very important here, I have just added this to show you the plumule and the radicle. Here you can see that the two key components of a germinating seed is the plumule and the radicle. Plumule is the upper component, okay, upper component that grows in the upward direction, and radicle is the lower component that grows in the downward direction. Okay, now this plumule, let me scroll it down. Plumule gives rise to the shoot system, whereas the radicle gives rise to the root system. Okay. So if this is your seed, we will have a plumule in the upward direction and radicle in the downward direction. Okay. This is plumule and this is radicle. Now this plumule will give rise to the shoot system because it will develop into a shoot, whereas the radicle will give rise to the root system because it will develop into a root, right? Now, these roots and shoots will gradually increase in length, okay? So, what is responsible for the increase in length of the root and shoot? These are the special type of meristematic cells known as shoot apical meristem and root apical meristems, okay? So we have shoot apical meristem SAM and root apical meristem RAM, right? What are the meristematic cells? These are the special type of cells which continuously divide and they lead to increase in the length or the width of the plants. Okay, so here we are talking about the increase in length of the plant, not the width. So you should also know that there are two types of meristematic cells: the primary meristematic cells and the secondary meristematic cells. Okay, secondary meristematic cells. The primary meristematic cells lead to increase in length of the plant. Okay, increase in length of the plant, whereas the secondary meristems lead to increase in the girth or thickness of the plant. Okay. So here shoot apical meristem leads to increase in shoot, increase in the length of the shoot, and root apical meristem leads to increase in the length of the root, right? So they both are leading to increase in length. That means that these two are the primary meristems. Primary meristems both are involved in increasing the length of the plant. Okay, why they are known as apical meristem? Because they are located at the apex. Okay, shoot apical meristem is located at the apex of the shoot and the root apical meristem are located at the apex of the root, right? Now another very important thing that you should remember is that these root apical meristems and shoot apical meristems are present in the immature part of the plant. Okay, these two are present in the immature part of the plant. Immature parts. Or we can say the developing parts of the plants. Developing parts. Okay, so these parts, that means the growing roots and growing shoots, are not yet developed fully or completely. These are in the process of development, right? So these shoot apical meristems and root apical meristem will be present only in the growing parts or developing parts, not in the developed parts, right? That is why if you take a small plant, okay, that is not mature enough, and you cut the stem of the plant, then the stem will grow back, right? Why they will grow back? Because they have the shoot apical meristems present in them, right? But if you take a fully developed tree, let's say this is the tree, and you cut the trunk of this tree which is fully developed and matured, then will the tree grow back? No, it will not grow back, because the meristems or the meristematic cells are not present in the trunk of a tree, because this is fully developed and they are present only in the developing parts or the immature parts. Got it? Now, here we know that there is always some exception, right? So we also have an exceptional type of meristematic cells which are present in the fully developed part of the plant. And what is that meristem? That meristem is known as intercalary meristem. So here you can see the diagram. We have intercalary meristems which are present in the grasses. Okay. So here you can see intercalary meristems are present in the zone of elongation of monocot stem. Okay, so here we had taken the example of a grown tree where the meristem was not present, right? So this tree is a dicot plant. This is a dicot plant, but the grass is a monocot plant. So in case of monocot plants, there are some meristematic cells known as intercalary meristems which are present in the matured parts. So even if you cut the matured part of the grasses, or if they are grazed by the herbivores, these matured parts will again grow. They will grow back, right? Because of the presence of intercalary meristem. So here we have discussed about three types of meristems: shoot apical meristem, root apical meristem, and the intercalary meristem. Okay, and these three meristems are the primary meristems, because all these three are involved in increase of length of the plant, not in the width or the thickness of the plant. Okay, we will also discuss the second type of meristems later. Okay, now let's come to the tissues. We will start with the tissues, because we know that the internal structure is made up of tissues. Okay, the cells are organized to form tissues, and then these tissues will be arranged in different forms. So what are tissues? Tissues are generally known as the group of cells which have common origin. Okay, so normally they are defined as the group of cells having common origin, structure, and function. Okay, so normally you will see that it is said that they have common origin, they have common structure or similar structure, and similar function. Now, about origin, it is always correct. All the cells of a tissue are having same origin, right? But about structure and function, it can be correct or it can be incorrect. That means the cells of a tissue can have same structure or they can have different structure. They can have similar function or they can have slightly different function as well. For example, if we take xylem, we know that xylem is a tissue which is involved in the conduction of water and minerals in the plants, right? Conduction of water and minerals. Now this tissue, the xylem tissue, is also made up of different types of cells. They can have, the xylems have tracheids, they have vessels, they have xylem fibers, they have xylem parenchyma, right? So all these cells have same origin, no doubt. But do they have same structure? No. Xylem fibers, xylem parenchyma, tracheids or vessels all are different in structure. Okay, we will also see the structure of these things, but here we know that they are different in structure. So it is not necessary that all the cells of a tissue will have same structure. Okay. Also, they have slightly different function as well as different efficiency. Okay, so different cells of a tissue can vary in efficiency. Some cells can be more efficient than the other cells. Okay, so they usually perform common functions. Okay, it is not necessary that they will always perform common function, but usually they have common function, that is the conduction of water and minerals, but they can have slightly different mode of action, or they can have different efficiencies. Okay, and obviously they can be similar or they cannot be similar in structure. We have seen the examples. Different cells of xylem and different cells of phloem also are different in structures. Okay, we will discuss these xylem and phloem later in the chapter. Now let's come to the different types of tissues. So the tissues are broadly classified into two types: meristematic tissue and the permanent tissue. Just now we have discussed about the meristematic tissues. What we have discussed? We have discussed that the meristematic tissues are present in the growing parts of the plants or in the immature parts of the plants, right? Whereas the permanent tissues are present in the fully matured or fully developed parts of the plants. So let's discuss further. So the tissues can be broadly classified into two groups. First one is the meristematic tissue and the other is permanent tissue. Okay, meristematic tissues contain the meristematic cells which divide continuously. We have just now discussed about the meristematic cells, that these are present in the growing or developing parts of the plants, right? And the developing parts of the plants need to grow continuously in length as well as in thickness, right? Therefore, these cells show continuous cell division, or the meristematic tissues show continuous cell division. All the cells are present in the meristematic tissues will divide continuously, which will lead to growth of the plant or development of the plant, right? Whereas the permanent tissues have lost the capacity of the cell division because these are present in the fully developed or fully matured part of the plants, which do not need to grow any further. Okay, so these cells will not be involved in the growth, but they will be involved in other functions of the plant, such as conduction of water and minerals or food. Okay, or any other function, right? For example, mechanical support. They can perform various other functions, but they are not involved in the growth. Therefore, they do not need to divide, right? And this is the reason why they have lost the capacity of cell division. Now let's first discuss about the meristematic tissues. Most of the things we have already discussed earlier in this chapter, right? We know that two types of growth are possible in plants. It can be primary growth or it can be secondary growth. Primary growth means what? Increase in length of the plant. And secondary growth means increase in girth or thickness of the plant. Okay, primary growth means increase in length of the plant. Now this increase in length can be due to apical meristem or intercalary meristem. Both of these we have discussed. Apical meristems are those meristems which are present at the apex of the roots or shoots. Okay, so if the apical meristems are present at the apex of the shoot and they lead to increase in length of the shoot, then these are known as shoot apical meristem, that is SAM. Okay, and when they are present at the apex of the root and lead to increase in length of the root, then they are known as the root apical meristem. So two types of apical meristems are there: shoot apical meristems and root apical meristems. Okay, which lead to increase in length of the shoot and root respectively. Now the third type of meristematic cells that we had discussed were intercalary meristems. Intercalary meristems were present between the matured tissues of the monocot plants such as the grasses. Okay, so they are present between the matured tissues of monocot plants, not the dicot plants. Okay, so if you cut the matured tissue of the monocot plant such as grass, they will grow back. But if you cut the matured part of a dicot plant, they will not grow back because the meristematic cells are not present in the matured parts of the dicot plants. In dicot plants, they are present only in the immature or the developing parts of the plant, such as growing roots or growing shoots. Got it? Then This was about the primary growth. What about the secondary growth? Secondary growth is also known as the lateral growth or the cylindrical growth, because the main stem of the plant is cylindrical. So the increase in thickness is the cylindrical growth or the lateral growth of the plant. Now here you have to remember an important term, that is cambium. Okay, the word cambium is very important here, because if the cambium is present, then only secondary growth will take place. If the cambium is absent, then the secondary growth will not take place. Now two types of cambiums are there, cork cambium and the vascular cambium. Cork cambium are present in the cork of the plant, and vascular cambium are present in the vascular bundles of the plant, such as xylem and phloem. Okay, so here you can see that the vascular cambium can also be of two different types, intrafascicular cambium and the interfascicular cambium. Okay, intra means in between the or within the vascular bundles, right? And inter means in between different vascular bundles. Okay, for example, if let me draw a diagram here. If this is your vascular bundle, this lower part is xylem, okay, and the upper part is phloem. This is the phloem, right? So if the cambium, vascular cambium, is present within this vascular bundle between the xylem and the phloem, let's say this is the cambium, then this is the intrafascicular cambium, because this is located within the vascular bundle. Now similarly, we can have another vascular bundle with xylem on the lower side and phloem on the upper side. So this is another vascular bundle. So if the vascular cambium is present between these two vascular bundles, then they will be known as interfascicular cambium. Okay, intrafascicular cambium was present within the vascular bundle between xylem and phloem, and the interfascicular vascular cambium is present between two different vascular bundles. Got it? And these cambium are responsible for the secondary growth in plants. We will discuss that later in the chapter. Right now, let's see the permanent tissues. Permanent tissues, they have lost the capacity of cell division, they do not divide. Now, again, permanent tissues can be of two types, simple permanent tissues or complex permanent tissues. We will also see the diagrams. Here we are just having the overview. Okay, so simple permanent tissues are those permanent tissues which have all the cells of same type. But the complex tissues are those which have different types of cells. Okay, so simple tissues, three types of simple tissues are there: parenchyma, collenchyma, and sclerenchyma. Okay, the next topic is the discussion of parenchyma, collenchyma, and sclerenchyma. Here just remember that two types of permanent tissues are there: simple permanent tissue and complex permanent tissue. Simple tissues or simple permanent tissues are made up of same type of cells, whereas the complex permanent tissues are made up of different types of cells. The examples of complex permanent tissues are xylem and phloem. Okay, we know that xylem have different types of cells such as tracheids, vessels, xylem fibers, and xylem parenchyma. All are of different types. Therefore, they come under the category of complex permanent tissues, right? Similarly, in phloem, we have sieve tubes, companion cells, right? All these are different types of cells, and therefore they are complex permanent tissues. Now, we will discuss them in detail as well. Okay. Meristematic cells we have already discussed at the beginning of the chapter. Now we have to discuss the permanent tissues in the detail. Okay. So first simple permanent tissues and then complex permanent tissues. Let's start with the simple permanent tissue. First we will have a look at the diagram, because if you understand the diagram well, then you will understand the topic very well. Okay. If you don't understand the diagram, then it will become confusing. Here we have the three different types of simple permanent tissues: parenchyma, collenchyma, and sclerenchyma. Now, what are the visible differences among these three types of simple permanent tissues? Parenchyma, you can see these are oval or round in shape, whereas the collenchyma are usually hexagonal in shape. Okay, these are not round, these are hexagonal in shape. They have 6 sides: 1, 2, 3, 4, 5, 6. So they are hexagonal in shape, right? Now Another difference is that in case of parenchyma, the cell wall appears to be very thin, whereas in collenchyma, the cell wall is thicker as compared to the cell wall of the parenchyma, right? In sclerenchyma, again, they have the thickest cell wall. Okay, so the thickness of the cell wall is increasing as we are moving from parenchyma to sclerenchyma. So here you also need to remember that the cell walls are made up of different things. For example, in parenchyma, the cell wall is made up of cellulose. Okay, so the parenchyma have cellulosic cell wall, whereas in collenchyma, the cell wall has cellulose, but in addition, it also has hemicellulose and pectin. Okay, so in collenchyma, the cell wall has cellulose, then it has hemicellulose, hemicellulose, and pectin as well. So these three things are present in the cell wall of the collenchyma. Therefore, the cell walls of collenchyma are thicker than the cell wall of the parenchyma, right? And not just thicker, they are also more rigid as compared to the parenchyma. Therefore, they can also provide mechanical support to some parts of the plants. Okay, whereas the parenchyma cannot provide mechanical support, right? Now, both parenchyma and collenchyma were living cells, whereas the sclerenchyma, the last type of simple permanent tissue, is the dead type of tissue. Okay, this is dead. The parenchyma and collenchyma were living tissues, whereas the sclerenchyma are dead, and they have thick lignified walls. That means the cell walls of sclerenchyma are made up of lignin. Okay. Now look at the structure so that you can remember the figure and then you can easily understand the concept. Okay, so just remember the main differences between the cell walls. The cell walls of the parenchyma is very thin, the cell wall of collenchyma is thicker, and then the sclerenchyma have thickest cell wall. What are the components of cell walls? In parenchyma it is cellulose, in collenchyma it is cellulose, hemicellulose and pectin, whereas in sclerenchyma it is lignin. Okay, now let's summarize the parenchyma. We have seen that the parenchyma cells are living and isodiametric. That means all the cells of the parenchyma are same in size. Okay, they are of same size. Then they have thin cellulosic cell wall. We have already discussed. They have very thin cell wall and they are made up of cellulose. So cellulosic cell wall. Then small intercellular spaces are present between the cells of the parenchyma. Okay, and the intercellular spaces are not present in the collenchyma or sclerenchyma. These are present only in the parenchyma, right? What are the functions of the parenchyma? They are involved in the synthesis of food. They can be involved in storage of food as well as secretion of materials. So these three functions of the parenchyma you should remember. Now another type of simple permanent tissue is collenchyma, which are also living in isodiametric like the parenchyma cells. They have central vacuole. Okay, so we know that the cells of the collenchyma are hexagonal like this, and they have central vacuole. So vacuole is present in the center, and the cytoplasm or the protoplasm is present at the periphery. So they have the peripheral protoplasm. Okay, the central space is occupied by the vacuole and the peripheral space is occupied by the protoplasm. This is the characteristic of collenchyma. Okay, and unlike the parenchyma, they have no intercellular spaces. In parenchyma, intercellular spaces were present. In the collenchyma, no intercellular spaces are present, which is another reason for them being more rigid than the parenchyma cells. Okay, why they are more rigid than the parenchyma? First of all, they have their cell wall has the deposition of cellulose, hemicellulose and pectin. Also, they have no intercellular spaces, which makes them more rigid than the parenchyma. Okay, what is the function of the collenchyma? Since they are more rigid, therefore they can provide mechanical strength to the young stems and petiole of the leaves. Okay, so they are rigid but not as rigid as compared to the sclerenchyma. Okay, they are the most rigid, right? Therefore, these collenchyma can provide mechanical support but only to the lighter part of the plants or weaker part of the plants such as the young stems or petiole of the leaves. whereas the sclerenchyma will provide the mechanical support to the complete part, even to the heaviest part of the plant, okay, such as the trunk of the plant. Now we come to the last one, sclerenchyma. These are dead at maturity. Okay, both parenchyma and collenchyma were living, but sclerenchyma are dead. Now, based on the difference in the shape, origin, and development, these sclerenchyma can be of two types. Now, here one question might arise that if these parenchyma, collenchyma, and sclerenchyma are simple permanent tissue, that means made up of same type of cells, then how we are saying that there are two different types of sclerenchyma? They must be having same type of cells, right? So two types of cells are possible, but at a time there will be only one type of cell present in the tissue. Either the sclerenchyma will have all the fibers or they will have all the sclerenes. Okay, two types are present. They can be fiber, they can be scleride. Okay, but both cannot be present in the same tissue because these are simple permanent tissue, not complex. So either they will have all the fibers or they will have all the sclerenes. Now the visible difference you can see here between a fiber and a scleride. Okay, the structure is entirely different. The fiber is long, thin and long cell, whereas scleride is round or oval in shape. Okay, there is a complete difference in the structure of these two cells. Again, here you can see that the fiber has a lumen. Here the lumen is also present in scleride. But one thing you can see that in scleride there are branches. Okay, branching is present in the scleride, which is not present in the fiber. Okay, and the scleride has numerous pits. A large number of pits are present in the scleride due to the branching. Okay, so let's see the characteristics of the fiber and the scleride. In fiber, you can see we have tapered end, tapering end, okay, which are like this. Okay, look at the diagram. Like this. Tapering end means like this, right? They have long, thin and narrow cells, right? As clearly visible from the diagram. Just like the hair which is present on our head, just like our hair, they are very thin and narrow. Okay. Then they have no protoplasm. Again, important point, the fiber has no protoplasm. Now let's come to the scleride, which are round or oval in shape. They show branching. Branching is the most important characteristic of the scleride. They have narrow lumen and they have numerous pits. Okay, so these characteristics you should remember. So this was all about the simple permanent tissues. How many types of simple permanent tissues were there? Three types: the parenchyma, the collenchyma, and the sclerenchyma. Okay, parenchyma and collenchyma were living, and the sclerenchyma is dead. Okay, collenchyma and sclerenchyma both can provide mechanical support, but collenchyma can provide mechanical support only to the weaker part of the plant, whereas the sclerenchyma can provide the mechanical support to the whole plant. Okay. Now we come to the complex permanent tissues. What do we mean by complex permanent tissues? Complex means they are made up of more than one type of cell. Okay, they are made up of different types of cells. Different types of cells. Two important complex permanent tissues are there, xylem and phloem. Okay, xylem is the complex permanent tissue which is involved in the conduction of water and minerals. Okay, it is involved in the conduction of water and minerals. Now from the name itself, it is clear that they will have different types of cells. So the different types of cells present in xylem are tracheids, vessels, xylem parenchyma and xylem fiber. Now parenchyma we have just now discussed. We know that the parenchyma are the living tissues. So the xylem parenchyma here are living, whereas all the other cells of the xylem are dead. Okay, so the tracheids are dead, the vessels are dead, and xylem fibers are also dead. We have also discussed about the fiber. These are dead cells. So in case of xylem, only the xylem parenchyma is living. All the other cells are dead cells. Okay. Here we have the diagram as well. Look at the diagram. You can clearly see that they have different shapes also. Tracheids, vessels, xylem parenchyma and xylem fiber. Out of all the 4 types of cells, only xylem parenchyma is living. Therefore, you can see a nucleus is present in the xylem parenchyma and it is not present in any other cells. Okay. Also, that cytoplasm is also present because it is living cell. Right. Here you have to remember another important terms, protoxylem and metaxylem. Proto means first and meta means later. Okay, so protoxylem are the xylem which are formed first or which are formed before, and therefore these are known as primary xylem. Whereas the metaxylem are also known as secondary xylem because they are formed later. Okay, these are formed after the protoxylem. Okay. The other type of the complex permanent tissue is phloem. Xylem was involved in the conduction of water and minerals, and phloem is responsible for the conduction of food materials, okay? Or the conduction of food particles or food materials, right? Again, this is a complex permanent tissue. That means it will contain different types of cells. So what are the different types of cells present in the phloem tissue? These are sieve tubes, these are companion cells, phloem parenchyma and phloem fibers, okay? Now, in case of xylem, we had seen that only xylem parenchyma was living cell, all the others were dead cells, right? But here only one will be dead and all the other will be living. Okay, so which one is dead? Phloem fiber is dead. Okay, because we know that the fibers which are type of sclerenchyma are dead cells. Okay, so these are phloem fibers are dead and all the others are living. So the sieve tubes are living cells, the companion cells are living, and phloem parenchyma are also living. Okay. Now here, why companion cells are known as companion cells? Companion means they will accompany the sieve tubes. They will help the sieve tubes. Okay. So these companion cells are closely associated with the sieve tubes, right? Because the sieve tubes do not have nucleus and the companion cells have nucleus. So if you look at the diagram given on the right side, let me scroll it down, then it will be visible. On the right side, we have a diagram. Here you can see the sieve tubes, this sieve tube, and these are the companion cells which are associated with the sieve tubes. You can see that the companion cells have nucleus. We know that the activities will be controlled by the nucleus. So who will control the activity of the sieve tubes because they do not have nucleus? So this will be done by the nucleus of the companion cells. Therefore, the companion cells are in close association with the sieve tubes. Therefore, this name is given to them. Okay. Now here one more important thing is related to xylem. In xylem, we have seen two different types of xylem, protoxylem and metaxylem, right? Protoxylem means primary xylem and metaxylem means secondary xylem, okay? So primary xylems are formed first and secondary xylems are formed later. So let me write it again here. Protoxylem, protoxylem, xylem is the primary xylem. That means it is formed first, and the metaxylem is formed later. Now if we draw diagram and we draw smaller circles for protoxylem and larger circles for metaxylem. Okay, suppose the smaller circles are showing the protoxylem and the larger circles are showing the metaxylem. So there are two types of the xylem based on the direction of formation of xylem. Okay, it can be endark xylem, endark or endarch xylem, and it can be exarch xylem. Okay, what do we mean by endarch xylem? When the formation of xylem starts from the center of the organ, let's say stem or root, and it moves towards the periphery, then it is known as endarch xylem. Okay, for example, if we take a transverse section of a stem, this is the transverse section of a stem. So in case of endarch xylem, the formation of xylem starts from the center. That means the protoxylem will be present at the center, right? The smaller sized These are present at the center. Whereas the later formed xylem, that means the secondary xylem, will be present towards the periphery. Got it? You can see that the smaller circles at the center means protoxylems are at the center. So the protoxylem have formed first, right? So the formation had started from the center and it has moved towards the periphery. Okay. So the later formed xylem or the secondary xylem are at the periphery, right? These are the secondary xylem, and the first formed xylem are at the center. Primary xylem or the protoxylem are at the center. This is known as endarched xylem, and this type of xylem is found in the stems. These are found in the stems. Whereas the other type of xylem is exarch xylem, which is found in the roots. So what happened in the exarch xylem? Here the protoxylem will be present towards the periphery. So if this is the transverse section of root, then here the protoxylem, that means the small size xylems, are present at the periphery. So what can we say from here? That the formation has started from the periphery, from the outside, right? And the later formed xylem will be present at the center. So here the primary xylem or the protoxylem is present at the periphery and the metaxylem or the secondary xylem present at the center. So this is the basic difference between the endarch and the exarch xylem. Endarch is found in the stems, exarch is found in the roots. Okay, we will also discuss this later in the chapter, but now I wanted to explain this to you. Okay. So do not confuse between the endarch and the exarch xylem. Now we come to the tissue system. Now tissue system, we know that different types of tissues are present in the plants. All these tissues are classified into three systems, three broader systems, depending on their location and the function. Okay, so different tissues will be located at different parts of the plants and they will also be involved in different types of functions. So on that basis, they are classified into three types of tissue system. First one is dermal tissue system, second one is vascular tissue system, and the last one is the ground tissue system. Okay, so first one is dermal tissue system. Dermal means what? We know, outer layers. Okay, outer layer. So the dermal tissue system forms the outermost covering of the whole plant body. Okay, it is forming the outermost covering of the primary plant body. Primary plant body. Okay, it comprises epidermal cells, stomata, and the epidermal appendages. So what are the components of the dermal tissue system? Epidermal cells, then stomata can be present in the leaves and young and the green stems, and epidermal appendages such as the root hairs or trichomes. Okay, so let's discuss them one by one. First component is epidermis, which is the outermost layer or the of the primary plant body. Okay, now we know that the epidermis can cover the outer layer as well as the inner lining. Okay, for example, if this is an organ, so the epidermis can be present on the outer layer, protecting it from the outer environment, as well as it can be present in the inner lining of the organ, protecting the inner lining of the organ. Okay, it is made up of elongated, compactly arranged cells. So which type of cells are present in the outermost layer? These are elongated cells. Okay, elongated cells. These are not circular cells. These will be elongated like this. Okay, and they are compactly arranged. That means they are tightly arranged. Okay, they are tightly linked to each other. It means that they will have no intercellular spaces. Okay, therefore they form a continuous layer. Okay, it means they have no intercellular spaces. Intercellular spaces, these are absent in the epidermal layers. Then the cells are usually parenchymatous, that means living cells. Parenchymatous cells are living cells with a small amount of cytoplasm lining the cell wall and a large vacuole. So vacuole is occupying a larger space, just like the collenchyma. In collenchyma also we had seen that the vacuole was present in the center and the cytoplasm was present at the periphery. Similarly, here also small amount of cytoplasm will be present at the periphery or the lining of the cell wall. okay, and there will be large vacuole. Here we have a diagram of cross section of a dicot leaf. Okay, you can see we have upper epidermis and lower epidermis. So two dermal layers are there. If this is the leaf, we take the transverse section or the cross section, then we will see that there is an epidermal layer on the top as well as at the bottom. Okay, so at the top there is upper epidermis, at the bottom there is lower epidermis, and the cells, you can see these are elongated cells. These are tightly formed, tightly arranged cells forming a continuous layer. Okay, now on the upper epidermis, you can see there is a cuticle layer. Okay, layer of cuticle. Now what is cuticle? Cuticle is a waxy layer. Okay, this cuticle is a waxy layer. Waxy layer which acts as the protective layer. Which acts as the protective layer on the epidermis. Okay, so what is the function of cuticle? It reduces the water loss. It reduces, the cuticle reduces the water loss. So it is present in the leaves to reduce the water loss as well as on the stems. Okay, but it is not present in the roots. Right, cuticles are not present on the epidermis of the root because roots have to absorb water from the soil. Okay, so if cuticle will be present, they will reduce the absorption of water as well. Right, therefore these are absent on the roots. So this was one component of the dermal tissue system, epidermis. Second component is stomata. We know that the outermost layer of the leaves also has stomata. Okay, stomata are very important, which are also present on the young stems. Okay, to carry out the photosynthesis. So what is the function of stomata? They regulate the process of transpiration and gaseous exchange. Okay. Then they consist of stomatal pore, guard cells and subsidiary cells. Okay. So stomata, or in general stomatal aperture, consists of three main parts. The central part is the stomatal pore. Okay. A small pore in the center. Right, then there will be guard cells. These are known as guard cells because they are guarding the stomatal pore. So there will be two cells on the sides which will be guarding the stomatal pore like this. Okay, and these guard cells, their inner wall, which is near the stomatal pore, will be thicker as compared to the outer wall, which is away from the stomatal pore. Okay, so this is thicker wall. Right, now these guard cells also vary in shapes in dicot and monocot plants. In case of dicot plants, the guard cells are kidney shaped, whereas in monocot plants these are dumbbell shaped. Okay, we will also see the diagram, don't worry. And these guard cells possess chloroplasts and regulate the opening and closing of the stomata. So based on the action of the guard cells, the stomata will be closed or opened. Okay, and then subsidiary cells. So outside the guard cells, you can see the subsidiary cells like this. So subsidiary cells are nothing but the modified epidermal cells. Okay. These are modified epidermal cells. And then again, outside the subsidiary cells, normal epidermal cells will be present. Okay. So let's have a look at the diagram of these stomata now. You can see the stomatal pore in the center. Look at the first diagram. Stomatal pore in the center. This pore, this shaded part, this is the stomatal pore. This is being guarded by two cells. These are guard cells which are kidney shaped. Right, on the left side we have dicot plant and on the right side we have monocot. Okay, so this one is monocot, this one is dicot. We know that in case of dicot plants, the guard cells are kidney shaped. So here you can see the kidney shaped guard cells having chloroplasts. These green colored dots are chloroplasts. On the right side, we have monocot plants. Here you can see the shape of the guard cells. These are dumbbell shaped, right? Like this. Like this. This is the shape of the guard cells in monocot plants, right? And this shaded portion is the stomatal pore. And then we have the subsidiary cells outside the guard cells, which are modified epidermal cells, right? So this was the diagram of the stomata. Now we come to the third component of the dermal tissue system, First one was epidermis, second one was stomata, and the third one is epidermal appendages, right? The epidermal appendages mainly root hair and trichome. So these are small hair-like structures which can be present on root or stem. If they are present on root, they are known as root hair. If they are present on the stems, then they are known as trichomes. Okay. So root hairs are very soft and they are unicellular. The main difference between them is that the root hair are unicellular, whereas the trichomes are usually multicellular. And the trichomes can be soft or stiff, whereas the root hair are always soft. The trichomes can be branched or unbranched, or they may be secretory. That means they can secrete certain organic substances. For example, tannins, gums, resins, etc. Okay. We have the diagram of epidermal appendages. Here you can see on the left side we have root hairs. On the right side we have trichomes. So you can see the branches in the trichomes. Okay, trichomes can show branching, right? Whereas the root hairs are unbranched. Root hairs are unicellular. Trichomes are usually multicellular. Okay. Now we come to the vascular tissue system. So what are vascular tissues? We all know these are xylem and phloem. Okay, xylem and phloem are the vascular tissues, and xylem and phloem together constitute the vascular bundle. Okay, so in one vascular bundle there is xylem and phloem both. Right, now here what we are going to discuss under this vascular tissue system, we will be discussing the arrangement or organization of xylem and phloem, because we are studying the chapter anatomy, and in anatomy we have to study or discuss the internal organization of the plants, right? So here we will see how these xylem and phloem are organized in different types of plants, that means monocot and dicot, or in different parts of the plants, root, stems and leaves. Okay, so based on the relative position of the xylem and phloem, these can be classified as radial or conjoint. Okay, radial or conjoint. We know that within a vascular bundle, there will be both xylem and phloem, right? But how these xylem and phloem will be located within that vascular bundle? For example, if this is the vascular bundle, here the xylem and phloem can be located on different radius. Let's say this is xylem on one radius and this is phloem on another radius. Okay, again xylem on one radius like this and phloem on another radius. So this type of the vascular bundle is known as the radial vascular bundle. Okay, radial, the word radial is related to radius, and the word conjoint is related to joint. Okay, so in case of conjoint, the xylem and phloem will be joined together, whereas in radius they will not be joined together, they will be present on different radius. Okay, so here we have the diagram. You can see in the first diagram we have the radial vascular bundle. You can see this brown colored part. Let me zoom in. This brown colored portion shows the xylem and this blue colored portion shows the phloem. Okay, so you can see that xylem is located on one radius, phloem is located on another radius. Again, xylem on one radius, phloem on another radius. So both xylem and phloem are present within the same vascular bundle, but they are not present on along the same radius. They are present along different radius and they are not joined together. So this type of vascular bundle is known as radial vascular bundle. Okay, now R for radial and R for root. So from here you can remember that radial vascular bundles are found in the roots of the plants. Okay, where they are found in the roots of the plants. On the other hand, in conjoint type of vascular bundles, they will be joined together. That means they will be along the same radius and they will be joined. So let's see the diagram of conjoint vascular bundles. See, in this diagram, the first one shows the radial vascular bundle. Radial, that means they are present on different radii. In the second and the third diagram, they are present along the same radius, right? This is the xylem in the lower portion. This is the vascular bundle, right? The lower portion consists of xylem. This is xylem. And the upper portion consists of the phloem. Okay. Now here you can see two diagrams. this one and this one. Okay, let's say this is diagram A and this is diagram B. In both these diagrams, A and B, you can see that the xylem and phloem are located along the same radius and they are joint, right? So these are conjoint type of vascular bundles and these are found in stems or leaves of the plants, okay? Now, what is the difference between the diagram A and diagram B? Can you see in diagram A, cambium is not present, whereas in diagram B, cambium is present. Now, what is cambium? Cambium is a special layer of cell, layer of cell that is required for secondary growth in the plant. Okay, required for secondary growth. We have already discussed earlier that if cambium is present in the plant, then secondary growth can take place. If cambium is not present in the plant or is not present in the vascular bundle, then secondary growth cannot take place. Okay. Also, we had discussed about the interfascicular cambium and intrafascicular cambium. Let's recapitulate. Intrafascicular cambium. Intrafascicular cambium means what? The cambium will be present within the vascular bundle. If this is the vascular bundle, this is xylem, this is phloem, then the cambium will be present within this vascular bundle in between the xylem and the phloem. Okay, so if this is the cambium, it will be present between the xylem and the phloem within the same vascular bundle. Okay. On the other hand, in interfascicular cambium, the cambium will be present between two different vascular bundles. Okay, so if this is one vascular bundle, this is another vascular bundle, this is the third vascular bundle, then the cambium will be present between two vascular bundles, not within the same vascular bundles. You remember this? We had already discussed about the intrafascicular and interfascicular. So in this case, we are looking at the diagram of which type of cambium, intrafascicular or interfascicular. We are looking at the intrafascicular because this cambium is present within the same vascular bundle between the xylem and the phloem. Okay. Now, what is the role of cambium? Secondary growth. If this cambium is present, then secondary growth can take place. Now, does the secondary growth take place in dicot plants such as mango or guava? Yes, in dicot plants, secondary growth takes place. It means that cambium must be present in dicot plants. Okay, so if cambium is present, it means it is a dicot stem. It is a dicot stem. It is not root because in root radial vascular bundle is present, okay, not conjoint. In these conjoint can be present in stems or leaves, right? Now, which type of stem? Dicot stem. In dicot stem, cambium will be present between xylem and phloem. Whereas in monocot stem, there is no need for secondary growth. Therefore, cambium will not be present in monocot stem. So, this diagram A is that of a monocot stem. Okay, this is the vascular bundle of a monocot stem because in monocot stem, secondary growth does not take place. So there is no need of cambium, right? Now, here you have to remember important terms such as open vascular bundles and closed vascular bundles. Okay, what do we mean by open vascular bundles? See, we know that the presence of cambium means it is open to secondary growth, right? The presence of cambium means the vascular bundle is open for secondary growth. That means this type of vascular bundle can be called as open vascular bundle. Okay, so this last diagram containing the cambium between xylem and phloem is known as open vascular bundle. Open vascular bundle. It is open for secondary growth, right? Whereas the earlier one, the first one, where cambium is absent, this vascular bundle is not open for secondary growth. This is closed for secondary growth. Therefore, this type of vascular bundle is known as closed vascular bundle. So let's summarize the difference between the open vascular bundle and the closed vascular bundle. Here we have the differences. You have already understood, but we'll just summarize them. Open vascular bundle is present in gymnosperms and dicot stems. Okay, so they can be present in gymnosperms. Gymnosperms, you can see some of the gymnosperm plants are very large and the secondary growth takes place in them, right? So there also open vascular bundle will be present, and in dicot stems definitely they will be present. Okay, whereas in the closed vascular bundles are present in monocot stems as well as in the leaves of the seeded plants. Okay, because the secondary growth is not required there. The second point is that the secondary growth is present in the open vascular bundle definitely because of the presence of the cambium, and the secondary growth is absent in the closed vascular bundle because of the absence of the cambium, right? The third point is intrafascicular cambium lies between phloem and xylem. So we have already discussed there is an intrafascicular cambium between xylem and phloem in the open vascular bundle, whereas the intrafascicular cambium will be absent in the closed vascular bundles. And the last point is that the phloem and xylem are not in direct contact, obviously, because in between the xylem and phloem cambium is present, therefore xylem and phloem are not in direct contact, whereas in case of closed vascular bundle cambium is absent, so the xylem and phloem will be in direct contact to each other. Got it? So this was about the vascular tissue system. Here you can recapitulate what we had discussed about the protoxylem and the metaxylem. What did we discuss about the protoxylem? Protoxylem means the xylem which was formed first. Okay, let's write it down here. Protoxylem, they are formed first. They are formed first. Therefore, these are also known as primary xylem. Primary xylem. And we had also discussed that metaxylem are the xylem which are formed later. Formed later. That means the metaxylem are formed after the protoxylem. Okay. And therefore these are known as secondary xylem. Secondary xylem. So in this diagram, the smaller circles represent the protoxylem and the larger circles represent the metaxylem. Okay. So two types of xylems are there, in dark xylem and in exarch xylem. Okay. In dark xylem means the formation of xylem starts from the center. Okay. That means the protoxylem will be found near the center of the stem, right? And the metaxylem will be found at the periphery. Okay, because the formation has started from the center towards the periphery. Whereas in exarch xylem, what happens? The formation of xylem starts from the periphery towards the center. That means the xylem which is formed first will be found near the periphery. So the protoxylem, small sized, these are found near the periphery, and the large sized metaxylem which are formed later are seen at the center. Okay, so this was the difference between endark and exarch xylem. We had also discussed that exarch xylem is found in roots, right? And endark can be found in stems. Okay, so this was all about the vascular tissue system. The last type of tissue system is the ground tissue system. How many tissue systems were there? Three different types of tissue systems: the dermal tissue system, the vascular tissue system, and the third and the last is the ground tissue system. So what comes under the ground tissue system? Everything that does not come under the dermal tissue system and vascular tissue system will come under the ground tissue system. Okay, so let's summarize. So the first point is that all the tissues except epidermis and vascular bundles will be coming under the ground tissue system, because epidermis comes under the dermal tissue system and vascular bundles, xylem and phloem, comes under the vascular tissue system. And all the tissues other than these will come under the ground tissue system. So it can consist of simple tissues such as parenchyma, parenchyma and sclerenchyma. We have discussed about the parenchyma, parenchyma and sclerenchyma, right? So all these come under the ground tissue system. Then parenchymatous cells are usually present in the cortex, pericycle, pith and medullary rays in the primary stems and roots. Okay, so here we will be discussing about the cortex, pericycle, pith and medullary rays in the next topic, where we will look at the diagrams of the anatomy. Okay. So in those diagrams, we will see the cortex, pericycle, pith, okay, all these things. So these also come under the ground tissue system because they contain the parenchymatous cells, okay. And these the leaves contain mesophyll cells. You have studied about the mesophyll cells. These are found in the leaves. We will also see the diagram of these mesophyll cells. So these also come under the ground tissue system. So in general, you can say that anything that did not come under the dermal tissue system and the vascular tissue system will come under the ground tissue system, okay. Now the tissue system is complete. We have come to the last topic of the chapter, that is anatomy of the dicot and monocot plants. Okay, here we will be discussing about the anatomy of roots of dicot and monocot plants, anatomy of stem of dicot and monocot, and anatomy of leaves of dicot and monocot plants as well. So we will start with the root of the plants. Okay, first dicot root and then monocot root. So basically we have to compare between the dicot and the monocots. Okay, so we will start with the root and then we will discuss the stem and at last we will discuss the leaves. Okay, so let's start with the dicot root. Here the diagram looks quite complex. Okay, so do not be scared looking at the diagram. It is very easy. You just have to remember a particular sequence that I'm telling you. Okay, so look at this flow chart. The outermost layer is the epiplema. Okay, we are moving from the outermost layer towards the center. Okay, so outermost layer on the plant body is the epidermis, right? So the epidermis in case of root is also known as epiplema. So this epiplema is nothing but the epidermis of the root. Epidermis of the root. So what is the outermost layer of a dicot root? It is epiplema. And this epiplema comes under which tissue system? It comes under the dermal tissue system, right? Comes under the dermal tissue system. Then what is the next layer? The next layer is cortex. So here look at this diagram. You can see the outermost layer is shown in dark green color. Okay, let me scroll it down. This outermost layer of the cell is shown in the dark green color, which also has root hair on it. Okay, so these epidermal appendages are also part of the dermal tissue system. So these are the root hairs. Mostly there are large number of root hairs in the dicot roots. Okay, here only two root hairs are shown, but more number of root hairs are present. Okay, so this dark green colored layer is the layer called epiplema, right? Now, as we move below this epiplema, we can see light green colored cells, right? These light green colored cells, these are the layers called cortex. These are the second layer after the epiplema, okay? And the cortex contains circular cells, round shaped, thin walled cells, and we know that these are the characteristics of parenchymatous cells. Okay, so they contain thin walled round shaped parenchymatous cells with intercellular spaces, right? And there is not just a single layer in cortex. In case of epidermis, we saw there was a single layer, but in cortex there are multiple layers. Okay, so there are large number of layers in cortex, right? So here look at the flow chart. The outermost is epiplema containing a single layer of cells. Then the next layer is cortex, which has multiple layers of cells. And which type of cells are present in cortex? Thin walled, round shaped parenchymatous cells with intercellular spaces. So this you have to remember, they have intercellular spaces. Then the next layer after cortex is endodermis. Actually, what is the characteristic of endodermis? This is in fact the innermost layer of the cortex. We have discussed that the cortex contains of multiple layers. Okay, so let me draw here. So these are the round shaped parenchymatous cells of cortex, right? Multiple layers are present. Now the innermost layer of the cortex is not round shaped, it is barrel shaped like this, box shaped, okay, barrel shaped. This is also part of the cortex, but it is slightly different from other cells of the cortex. So this layer is known as endodermis. Endodermis. A different name is given to the innermost layer of the cortex because this layer is slightly different from other layers of the cortex. Okay, what are the differences? First, the difference is in shape. The other layers contain circular round shaped cells, but this layer contains barrel shaped cells. Also, we have seen that the other cells of the other layers have intercellular spaces, but these cells in the endodermis, they do not have intercellular spaces. They are tightly packed cells. So the cells of the endodermis are barrel shaped without any intercellular spaces. Okay. And the third important characteristic of endodermis is that this layer has a deposition of material known as suberin. Okay, suberin is deposited on the endodermis and this is known as Casparian strips. So if it is asked that Casparian strips are part of which layer of the dicot root, they are part of the endodermis. Okay, they are not part of the epiplema, they are not part of the cortex, they are part of the endodermis only. Okay, then the next layer that comes after the endodermis is pericycle. So if you look at this diagram, dark green color shows the epidermis layer, light green colors show the cortex layer and the endodermis layer, right? And in brown colored you can see pericycle is shown. Okay, so you can see brown colored ring like structure. This is the layer known as pericycle, which comes below the endodermis. Okay. And the next one will be the vascular bundles. Now, vascular bundles means xylem and phloem. So, in fact, the xylem and phloem or the vascular bundles are found within the pericycle region itself. Okay. So, you can see these are the xylem patches, right? Xylem bundles are shown in red color, right? Now, here you have to pay attention at the diagram. Can you see the protoxylem and the metaxylem written here? We have discussed that in case of roots, exarch xylem is found. Exarch xylem means the formation of xylem starts from the periphery towards the center. Okay, so it starts from the periphery and it moves towards the center. That means the xylem which are formed first or the protoxylem will be found towards the periphery. So here you can see that the protoxylem are found near the periphery. Okay, or near the pericycle, whereas the metaxylem or the later formed xylem are found near the center. Okay, so this you have to remember. This is known as exarch type of xylem. Exarch. We have discussed that exarch type of xylem is found in the roots. Okay, the case will be just opposite in stems of the plants. Okay, there in dark xylem will be found, not exarch. Okay. So what are the layers that we have discussed? Outermost is the epidermis, right? And the epidermis of the roots are known as epiplema. The next multiple layers are known as cortex. The innermost layer of the cortex is known as the endodermis, which contains the Casparian strips. Then after endodermis comes the pericycle, right? Pericycle layer, which are shown in brown color. Then the next one is the vascular bundles, xylem and phloem. So here you can see the exarch type of xylem is found in roots, right? And then at last we have the central part that is known as pith. Okay, so the central portion or the central cells are known as the pith. Okay, so pith is generally, we can say this is the center. This is the center. So remember, just remember this sequence, particular sequence: epidermis, then cortex, then endodermis, then pericycle, vascular bundle, and pith. If you remember this sequence, then this topic will become very easy to you. Okay. Now let's move to the next type of root, that is monocot root. Okay, you have understood the dicot root. Now we come to the monocot root. We basically have to compare the dicot root with monocot root. Okay, so most of the things will be same. For example, if you look at this diagram, I am zooming this diagram. This is the diagram of a monocot root. So can you see the outermost layer is the epiplema, this dark green color, this is epiplema, similar to that of the dicot root. Okay, so this is same. It also contains root hair, but the number of root hairs will be less in the monocot roots as compared to the dicot roots. Okay, so these are less than dicot roots. Then below the epiblema comes the multi-layered cortex, which contains the parenchymatous cells. Okay, parenchymatous, and they will have intercellular space. The innermost layer of the cortex is the endodermis, same as that of the dicot root. So these things are common, right? Endodermis is the innermost layer of the cortex having barrel-shaped cells. Barrel-shaped cells without any intercellular spaces. Okay. Then again we have the pericycle. Okay, this brown colored layer shows the pericycle. This is the pericycle. Brown colored layer is the pericycle. Then we have the vascular bundles, and then we have the pith. Okay, pith is the central part. Pith is the central part. Now, what is the difference between the vascular bundles of the dicot root and the monocot root? In case of dicot root, the number of xylem bundles were generally less. Here you can see there are more number of xylem bundles. You can see these red colored structures. These are the xylem bundles, and these yellow colored in between the xylem bundles, these are the phloem. Okay, so xylem and phloem are present. You can see these are present along different radius. We have also discussed about the radial and the conjoint type of vascular bundles. So we saw that in case of roots, which type of vascular bundles are found? Radial vascular bundles. That means xylem and phloem will be found on different radius. Therefore, you can see the red colored xylem is on one radius and the yellow colored phloem is on another radius. Okay, this is radial type of the vascular bundles. Okay, so we had to compare the dicot root with monocot root. So what is the comparison? We concluded that the number of xylem bundles are more in monocot roots as compared to that in the dicot roots. Okay. Also, the pith is larger. Okay, the pith is larger here. In dicot, the pith was very small. Okay. So let's summarize now. I hope you have understood the diagram. Most of the things are common. Okay, most of the layers are common between the dicot and the monocot roots. We will only focus on the differences. So the first point is that in monocot roots there are more than 6 xylem bundles. So this is known as polyarch xylem. Poly means many. So there are many xylem bundles. So polyarch. Whereas the number of xylem bundles was less in dicot roots. Second point is the pith is large and well developed. Okay, so the pith of the monocot root is larger and more well developed as compared to that of the dicot root. And the last point is do not undergo secondary growth. Obviously, we know that the monocot plants do not undergo secondary growth, so the cambium will be absent. Okay, whereas cambium will be present in the dicot. So here I don't know if cambium is shown in the diagram of dicot roots or not, but it will be present because this is the dicot plant, and in dicot plant secondary growth takes place. Okay. So I hope you have understood the difference between the dicot root and the monocot root. Similarly, we will study about the stem and we will find out the differences between the dicot stem and the monocot stem. Okay. So first we will have a look at the dicot stem and then we will compare it with the monocot stem. Okay. You remember the sequence of the layers of cells? Outermost layer is epidermis. Okay. So the epidermis was the outermost layer in the root and it will be the outermost layer in the stems as well. Okay. The only one difference is that in case of roots, the epidermis is also known as epiblema. Okay. Here we will simply call it epidermis. Right. Now we know that the epidermis of stems and leaves can have thin layer of cuticle. We have also discussed earlier in the chapter about the cuticle. It is a layer of waxy substance. Okay. So it is a waxy layer which is useful in reducing the water loss. It reduces the water loss. Okay, the cuticle layer is not present in the roots because the roots have to absorb water from the soil. Okay, now this was the outermost layer, and similar to the roots which have root hairs, the stems have trichomes. Okay, so the hairs present on the stems are known as trichomes, and these can also have few stomata. Okay, so a small number of stomata can also be present on the epidermis of dicot stem. Okay, then the next layer after epidermis is the cortex. Now here the cortex region is very important, right? We know that in case of roots, the roots are very thin, right? Whether it is dicot plant or monocot plant, the roots have to be very thin, but the stems have to be thicker than the roots. Okay, that means the stems will have additional layer of cells, right? So although epidermis, cortex, pericycle, pith, these will be common in roots and stems, but in stems there will be an additional layer which are known as hypodermis. Okay, so hypodermis are actually part of the cortex itself. This is the first layer of the cortex. Okay, just like the endodermis, which was the last layer of the cortex, hypodermis is the first layer of the cortex. Okay, so here in case of dicot stem, the cortex can be divided or classified into three zones: zone 1, zone 2, and zone 3. Okay, so zone 1 contains the layer known as hypodermis, which contains collenchymatous cells. Okay, so we have discussed in the roots that the cortex region contains parenchymatous cells, right? But the hypodermis layer of cortex in case of dicot stem contains collenchymatous cells. What are the characteristics of collenchymatous cells? These cells are hexagonal in shape. Okay, these are hexagonal in shape, whereas the parenchymatous cells are round shaped, circular shaped. Okay, collenchymatous are hexagonal in shape. Another important thing is that the collenchymatous cells have cell walls composed of cellulose, hemicellulose, and pectin, which makes the cell wall of collenchymatous cells thicker than the cell walls of the parenchymatous cells. Okay, and they also do not have the intercellular spaces which are present in the parenchymatous cells, right? So the hypodermis of the cortex contains collenchymatous cells. This you have to remember. Also, when we will compare the dicot stem with the monocot stem, you will see that the hypodermis of the monocot stem is different. Okay, that contains sclerenchymatous cells, whereas the hypodermis of the dicot stem contains collenchymatous cells. This is very important difference. Okay, the second zone consists of the normal parenchymatous cells which were also present in the roots. Okay, so these contain rounded thin walled parenchymatous cells, and the last zone, zone 3, contains the endodermis, which is the innermost layer of the cortex, and this endodermis is rich in starch grains, which is known as starch sheath. Okay, so starch sheath is part of the endodermis, right? Then the next layer after cortex is pericycle. Pericycle layer, again, it is present in the form of semi-lunar patches of sclerenchyma. This pericycle of the dicot stem is also different from the pericycle of the roots. Okay, although the name is same, but the constituents are different. Okay, components are different. Here the pericycle is present in the form of semilunar patches of sclerenchyma. So in dicot stem we have parenchymatous cells, we have collenchymatous cells, and we have sclerenchymatous cells as well. All three types of cells are present in dicot stem. Okay, pericycle has sclerenchymatous cells. Then the next layer is vascular bundles, that is xylem and phloems, which were also present in the roots. But the most important characteristic of the dicot stem is that the vascular bundles, that means xylem and phloem, are present in the form of ring. Okay, they are arranged in the form of ring. Okay, they have ring arrangement. Let's have a look at this diagram. Okay, this is the diagram. I hope it is clear to you. This is the diagram of the transverse section of dicot stem. Can you see the epidermal hair outermost on the outermost layer? Okay. On the right side also we have the outermost layer, epidermis. Below the epidermis we have cortex region. Okay, cortex region from here to here in both on both sides. You can see the outermost layer of the cortex is hypodermis, which contains collenchymatous cells. Here you can see collenchyma are present in the hypodermis. After that, we have normal parenchymatous cells in the cortex, and the innermost layer is the endodermis. Okay, so this region is the cortex region. Cortex region containing hypodermis, then normal parenchymatous cells, and then endodermis. Then below the cortex comes the pericycle. Okay, pericycle. Then we have the vascular bundles which are arranged in the form of ring. Here you can see these are the vascular bundles. Vascular bundles. This is vascular bundle. This is vascular bundle. They are arranged in the form of a ring. So this ring arrangement is the characteristic feature of dicot stem, which is not found anywhere else. Okay. In monocot stem also they are not present. They are not arranged in the form of ring. They are scattered in monocot stems. Okay. Then we have the medullary rays. So what are medullary rays? These are normal parenchymatous cells which are radially present between the in between the vascular bundles. So these are the vascular bundles. In between the vascular bundles there will be few layers of radially placed parenchymatous cells. Okay, and these are known as rays, medullary rays, because they look like rays of the parenchymatous cells present radially in between the vascular bundles. Okay, and then the innermost or the central part is the pith, which contains large number of parenchymatous cells. Okay, so I hope you have understood the diagram. You can see protoxylem and metaxylem. Here the metaxylem is present near the center. Okay, and the protoxylem is present near the periphery. It means it is which type of xylem? Endark or exarch? It is endark xylem. Endark xylem. Okay. I have said opposite. I have said that protoxylem is towards periphery, which is not the case. I'm sorry for that. The protoxylem is located towards the center and the metaxylem is located towards the periphery. Okay, this is the lower part is the center and the outer part is here like this. Okay, this is inner part. This is outer part. So in the inner part, protoxylem is present and in the outer part, the metaxylem is present. That means the formation of xylem has started from the center. So this is endark type of xylem. And we know that in case of stems, endark xylem are found. Whereas in case of roots, exarch xylem are found. Okay. So if you have understood the concept, it will be easy. Otherwise, it will become very confusing. Okay, because so many terms are there. I hope you understood the diagram well. Innermost part is pith, which contains a large number of parenchymatous cells. Okay. And here an important point is given. Vascular bundles of dicot stems are conjoined. Conjoined means the xylem and phloem are joined together. They are present on the same radius like this. This is xylem and this is phloem. Okay, they are present on the same radius. Therefore, these are known as conjoint. They are not present on different radius, which is the characteristic of roots. Okay, in roots, they are present on different radius. So, the xylem, the vascular bundles are radial in case of roots and conjoined in case of stems. Okay, also, the vascular bundles are open. Open means what? Open for secondary growth. That means cambium is present. So, here the cambium is present between the xylem and the phloem. And the cambium will be absent in monocot stem because monocot stem do not undergo secondary growth. It will be present only in the dicot stem. Okay. With endarked protoxylem. Endarked protoxylem means the xylem, the formation of xylem starts from the center, right? So the protoxylem will be present near the center and the metaxylem will be present towards the periphery. Got it? So this was all about the dicot stem. If you have understood it well, then monocot stem will be very easy for you. You just have to remember certain differences between the dicot and the monocot stem. Okay. We have seen that the most important characteristic of dicot stem is the ring arrangement of the vascular bundle. But in monocot stem, there is no ring arrangement. Instead, they are scattered throughout. Okay, so here you can see that a large number of vascular bundles are scattered throughout the stem. Okay, so large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath. So this is again important point. These are surrounded by the sclerenchymatous bundle sheath. Okay. Another important difference is that hypodermis, which is the first or the outermost layer of the cortex, is sclerenchymatous in case of monocot stem. In case of dicot stem, it was collenchymatous, but in case of monocot stem, it is sclerenchymatous. This is another very important difference. The first difference is that in dicot stem, the vascular bundles are arranged in the form of ring, whereas in monocot, they are scattered. Second important difference is the hypodermis of the dicot stem was collenchymatous, whereas the hypodermis of the monocot stem is sclerenchymatous. This is very important difference. You must not forget these differences, okay? Then other lines are same as given in the NCERT. A large conspicuous parenchymatous ground tissue is present. Vascular bundles are conjoined and closed. In case of dicot stem, the vascular bundles were open because cambium was present. Here, the cambium is absent. Cambium is absent, therefore it is closed. The vascular bundles are closed because there is no need of secondary growth in the monocot stem. Okay. Another important point which is given in the NCERT also, peripheral vascular bundles are generally smaller than the centrally located one. Just remember this point. Okay. Then the last point is phloem parenchyma is absent and water containing cavities are present within the vascular bundles. Okay. So this was all about the dicot and monocot stem. I hope the root and stem both are clear to you. Now we come to the next and the last topic, that is leaf. Okay, first we will discuss the dicot leaf, then we will compare it with the monocot leaf. So dicot leaf. Dicot leaf are also known as dorsiventral leaf. Dorsiventral is made up of two words, dorsal and ventral. So the dicot leaves are dorsiventral. It means the dorsal surface and the ventral surface of the leaf are different. Okay, we know that a leaf has two surfaces, the upper surfaces and the lower surface, right? So both the surfaces are different, they are not same. So what is the main difference between the upper surface and the lower surface? The main difference is in the number of stomata. Okay, the number of stomata is more on the lower surface as compared to that on the upper surface. Okay. In fact, sometimes the upper surface may even lack stomata totally. There can be no stomata on the upper surface, but on the lower surface, a large number of stomata will be present. Okay. Now, if you see a vertical section through the lamina of a leaf, you can see three main parts in the leaf. The first part is the epidermis, which is the outermost layer. Epidermis is also present in roots and stems. So the outermost layer is also present in the leaves. This is the epidermis with a conspicuous cuticle. What is cuticle? We have discussed cuticle is a layer of waxy substance. Okay, it is a waxy layer which reduces the water loss. It is present in leaves and stems but absent in roots because root has to absorb water from the roots, from the soil. Now the epidermis can be adaxial epidermis or abaxial epidermis because a leaf has two surfaces, the upper surfaces and the lower surfaces. So the epidermis will also be present on both the surfaces. So the epidermis which is present on the upper surface is known as adaxial epidermis, whereas the epidermis which is present on the lower surface is known as the abaxial epidermis. Okay. We have also seen that the lower surface will have more number of stomata as compared to the upper surface. Upper surface can even lack stomata totally. So this was the first part. And this epidermis is part of which tissue system? Dermal tissue system, right? The next part is mesophyll. Mesophyll is part of which tissue system? It is part of the ground tissue system, okay? Ground tissue system. Ground tissue system, because the mesophills are neither part of the dermal tissue system nor part of the vascular tissue system. So obviously they are part of the ground tissue system. And these are present between the upper and the lower epidermis. Okay, they contain chloroplast and carry out photosynthesis. They are made up of parenchyma. So the mesophills are parenchymatous. That means they are made up of parenchymatous cells, right? And parenchyma can also be of two different types, palisade parenchyma and spongy parenchyma. Now let's have a look at this diagram of the leaf to understand it well. We know that there will be epidermis present on both the surfaces, the upper surface as well as lower surface. So this is shown in the pink color. This pink color layer on the upper surface is the adaxial epidermis, and the pink colored layer on the lower surface is the abaxial epidermis. Right, this you have understood. Now in between the adaxial epidermis and the abaxial epidermis, a large number of green colored cells can be seen. These are the mesophyll cells. Okay, so these green colored cells are the mesophyll cells. But can you see that these mesophyll cells are different on the upper surface and the lower surface? On the upper surface, the shape is elongated, but on the lower surface, the shape is round or circular. Right. Also, on the upper surface, the cells are tightly packed, whereas on the lower surface, these are very loosely packed. Right, and there are air cavities present between the cells on the lower side which are absent on the upper side. Why is it so? The reason is that on the lower surface we have stomata present which are absent on the upper surface, right? So since stomata are present on the lower surface, there is a need for air cavities. So to make spaces for air cavities, such spongy mesophyll cells are present on the lower surface so that air cavities are present. On the upper surface, there is no need for air cavities, so the cells are elongated and tightly packed, and these are palisade parenchyma cells or palisade mesophils. So two types of parenchyma cells are found in the leaves, palisade and spongy. Palisade are found on the upper surface and the spongy parenchyma are found on the lower surface. Okay. Now let's move further. Here we have the differences between the palisade parenchyma and the spongy parenchyma. The palisade parenchyma are present near the upper surface, so these are adaxially placed. Spongy parenchyma are present near the lower surface, so these are abaxially placed. Or we can say that the spongy parenchyma are present below the palisade parenchyma. Then the second difference is in the shape. The palisade parenchyma are elongated cells and they are arranged vertically parallel to each other, and they are arranged very tightly. So they are arranged like this. Elongated cells placed vertically parallel to each other like this. On the other hand, the spongy parenchyma are oval, round and loosely packed cells like this. Got it? And since the palisade parenchyma are tightly packed, therefore no air cavities are present, but spongy parenchyma are very loosely packed. Therefore, numerous large spaces and air cavities are also present between the cells. Okay, so this was about the mesophils. Mesophyll is part of ground tissue system. And the last part of the leaf is the vascular system, which is nothing but the xylem and phloem, which are used for the conduction of water, minerals and nutrients. Okay. So these include vascular bundles which can be seen in the veins and the midrib. Why they are seen in the veins and the midrib? Because veins and midrib of the leaves are the pathways for the conduction of water and minerals, and vascular system also performs the same function, conduction of water and minerals. Therefore, these vascular bundles are found in the veins and the midrib. Okay, the size of the vascular bundles depend on the size of the veins. We know that in a dicot plant, suppose this is the leaf of a dicot plant, the size of the veins vary, okay, or thickness of the veins vary. This is the midrib. This is the midrib of the leaf. Midrib is thickest among all the veins. Okay, so this is the thickest. So it will have largest number of vascular bundles. It will have largest size of, sorry, not number, largest size of vascular bundles. Then the veins coming out from this midrib will be thinner as compared to the midrib. So it will have smaller size of the vascular bundles. Again, the veinlets coming from these veins will be even thinner. So they will have smallest size of the vascular bundles. So as the thickness of the veins and veinlets change, the size of the vascular bundles also change. Okay. And which type of venation is seen in dicot plants? Reticulate venation. Reticulate venation means the veins and veinlets have varying thickness. They are not of the same thickness. They are varying thickness. Okay. And vascular bundle is surrounded by a layer of thick-walled bundle sheath cells. So this also you have to remember. Vascular bundles are surrounded by the bundle sheath cells in the dicot leaves. Okay. Now, monocot leaf. The dicot leaf was also called dorsiventral leaf, whereas the monocot leaf are also known as isobilateral leaf. Iso means same, bi means two. So in case of dicot leaf, both the surfaces were different. Okay, there were differences in the upper surface and the lower surface of the leaf. Whereas in case of monocot leaf, there will be no difference in the upper surface and the lower surface of the leaf. Both the surface will be similar. Okay. That means stomata will be present equally on both the surfaces, unlike the dicot leaves. Okay. So the first point here is stomata present on both the surfaces of the epidermis equally. They can be slight change, but almost they will be equal in numbers. Now the second point is mesophils are not differentiated into palisade and spongy parenchyma. So in case of dicot leaf, we had two types of mesophil cells, palisade parenchyma and the spongy parenchyma. But here we know that the stomata, number of stomata are same on both the surfaces. So it means that on both the surfaces, there is a need for air cavities, right? In case of dicot leaves, on the upper surfaces, stomata were absent, or even if they were present, they were very few in numbers. So there was no need for air cavities. That is why palisade parenchyma was present on the upper surface. But here, there is a need for air cavities on both the surfaces because the stomata are present equally on both the surfaces. Therefore, only spongy parenchyma will be present which are loosely arranged. Okay, and these loosely arranged spongy parenchyma cells will allow spaces for air cavities. Right. Therefore, the mesophils will not be differentiated into palisade and spongy parenchyma. Only spongy parenchyma will be present on both the surfaces. Palisade parenchyma will be absent. Then, one important characteristic of monocot leaf is the presence of bulliform cells. Okay, bulliform cells. Bulliform. What are bulliform cells? These are large, colorless, and empty cells. Large, colorless and empty cells which are nothing but the modified adaxial cells. Okay. So bulliform cells are the modification of certain adaxial epidermal cells. These are large, colorless and empty cells. Why they are empty? Because they have to fill water in themselves. So initially they are empty. Now, what is the function of bulliform cells? So they help in indicating the water status of the plant. Okay, if water is available in sufficient amount, then these bulliform cells will fill water in themselves and they will become turgid. This will show that there is enough water or sufficient water. Therefore, leaf surfaces will get exposed. Because there is no need to conserve water. There is sufficient water available. So the leaf surfaces will get exposed. On the other hand, if there is scarcity of water, then these bulliform cells will become flaccid. They will not fill water in themselves, they will become flaccid. This will indicate water scarcity. Therefore, the leaves will curl inwards to minimize the water loss, okay, or to conserve water. So this is the role of bulliform cells which are found in monocot leaves, not in the dicot leaves. And then which type of venation is found? Parallel venation. In dicot leaves, reticulate venation was found. That means all the veins and veinlets had different thickness. But here in parallel venation, only the midrib will be the thickest. It will be thickest, but all the veins coming out from this midrib will be same. Will be of the same thickness. This is parallel venation and they will run parallel to each other. In case of dicot, there was no parallel venation. There was reticulate venation. There was no fixed direction for the veins. Okay, they were moving in any direction, but here they will have a fixed direction and all the veins will be parallel to each other and they will be having same thickness. Okay, so similar sizes of vascular bundles will also be present in all the veins because the size of the vascular bundles depend on the thickness of the veins. So here, except in the main veins, the size of the vascular bundle will be same. Okay, in the main vein, the size of the vascular bundle will be larger because the main vein is the thickest, right? Whereas all the other veins are of similar size, so they will have similar size of vascular bundles. Here we have the diagram for monocot leaf. You can see, have a look at the diagram. I hope you have understood it well. I've explained everything. Adaxial surface, abaxial surface, stomata is present on both the surfaces. We have mesophils containing spongy parenchyma. You can see here we have same type of parenchyma cells on both the surfaces. From upper to lower surface, all the cells are of same type. Then we have vascular bundles here. I hope you have understood this chapter well. Here we complete this chapter, anatomy of the flowering plants. If you have any doubt, you can mention that in the comment section. Okay. We will be back with another session, that is structural organization in animals, very soon. Till then, keep learning.